All right, we are on the last lesson of chapter 14. Um, and this one, we're going to be discussing the human genome. So because this is the last lesson, I highly recommend that if you haven't started your review yet, you kind of work on that. Don't wait till the last minute, because if you end up with questions, I may not get back to you in time. All right, so let's get started with how we've learned about the human genome. So the first step that we take when we're studying DNA and stuff, one of the first things that we'll do is manipulate the DNA. Okay, so to manipulate it, we need to be able to cut it, separate it, and then replicate the DNA base by base to read any of the sequences, read any sequence from any cell. So we need different methods to do each of these. Okay, so um, the first step is to cut it. The way that we cut it, we don't have like teeny tiny scissors so we can go into the cell and like cut the DNA. We're going to instead use what's called a restriction enzyme. Restriction enzymes are very, very specific substances, and they cut the DNA at a very specific location. Okay, so what they look for, usually what they're looking for are palindromes. So if you don't remember what a palindrome is, it's something that is read the same forwards and backwards, like the word race car. Okay. Um, and so they're going to cut at these very specific nucleotide sequences. And there are many different restriction enzymes. They all cut at their own types of, or their own locations. So they all cut at different regions, different sequences. So this is one example of a restriction enzyme. This one is called EcoRI. And so it looks for the sequence CTTAAG. And it cuts between the A and the G, and then it cuts across and cuts between the A and the G here. And so it creates these, what we call sticky ends, because it goes across. So a sticky end is useful when we want to do like genetic engineering. Genetic engineering usually involves like taking DNA from one thing and putting it in another thing. So one thing that they have done with that is taken a glow-in-the-dark gene from jellyfish, and you can insert that gene into bacteria, and then you have glow-in-the-dark bacteria. So the actual like function of that, like a real purpose behind that, is there isn't one except to be able to say, hey, we can put these genes from here over there. And actually, like, helpful method of that is they can take insulin genes. So they can take the genes for insulin to make insulin out of people. So they take a copy of like a human insulin gene, and they can insert that into bacteria and produce insulin. Because sometimes people don't make insulin for themselves, or their body doesn't respond to it correctly. So these sticky ends will let you, then you can cut out a gene and then put it back in somewhere else. That's really more than what we're talking about today, but that's why sticky ends are helpful, or why they are useful. Other restriction enzymes will create what's called a blunt end, where instead of being jagged so you can put it back together, it's just straight down. So you just have this piece of DNA that just, you can't really, it's still useful for our purposes with this. You could still cut your DNA into different pieces and then do the rest of what we're about to talk about. But you couldn't like cut a piece of DNA and have it with blunt ends and insert it into another sequence of DNA. That doesn't work. So anyway, restriction enzymes, they cut your DNA into very precise pieces. So those pieces are called restriction fragments. So here we have all these three DNA fragments. So each one would be a restriction fragment. Okay, and they are very, very specific. They cut at a very specific nucleotide sequence. The next step is to separate the DNA. The main way that we do this is using gel electrophoresis. So This process, you literally have it gel, like it's almost like jello, um, but it's not edible. Okay, but it has that same consistency to it. So you take your gel and you would put it down in, it's actually down in a liquid. And then up at the top, you have what are called wells. So it's essentially like a little hole or a notch in the gelatin, or in the gel. And that's where you would place your DNA that has been cut with your restriction enzymes. And then you put it down in a liquid that's called a buffer, and you attach electricity to it. So you have a positive and a negative pole. DNA itself is just very slightly negative. So by attaching the negative electrode to the end where you loaded your DNA, okay, where you put the DNA in, and then the positive electrode to the other end of the gel, you create this flow of electricity and it pushes the DNA toward the positive terminal. The smaller the fragments are, the more easily they can move through the gel. And so the smaller ones travel a lot farther and the longer ones stay pretty close. Okay, so we can use this process to actually read the DNA. And the way we do this is that we take just a single strand of DNA, so half of a DNA molecule. We put it in a test tube with DNA polymerase and a lot of bases. And some of those bases have a dye molecule attached and they're color coded. Okay, so for instance, the thymine bases some of them would have a yellow dye, and some of the guanine would have a red dye, and some of the adenine would have the green dye, or whatever. So that's the example they give here. But these dye molecules prevent another molecule from being, or another base from being added because they're too big. And so they just kind of get in the way. So anytime a dye labeled base is added, we stop DNA synthesis. So you get these DNA fragments of different sizes because they might stop after a couple of bases. They might stop after like 6 or 8, or they may not stop for, a whole bunch of fragments, or sorry, a bunch of bases. Okay, so we get these DNA fragments that have different sizes to them. And again, we can do electrophoresis and separate them based on their size. And then we read from the bottom, or from the furthest that it traveled, we read those colors because they each correspond to a DNA or a base. And so the furthest ones are the smallest, and then we would just go up from there and decode this, and we can read the whole sequence of DNA. Okay, so the ordered color of those bands is telling you what the sequence of DNA is in those fragments. Okay, so that's one way that we can read the DNA. The Human Genome Project took like between 10 and 15 years. I don't remember how long it actually took. I think that's right. And it was, it involved scientists from several countries working for several years, trying to sequence all of the base pairs in the human genome and identify all of the genes. So they broke the genome into smaller manageable pieces, and everybody was kind of responsible for their particular portion. And then one of the things they would do is identify very specific sequences throughout the genome to use as a marker. So that they could tell, okay, in relation to this sequence, where are we at on the chromosome? Okay. Kind of like mile markers, right? I assume you guys know what mile markers are on the highway. They tell you, okay, you're at mile, 137. They correlate, by the way, to the exit numbers, usually. Okay. Um, so that way you know where you are. So if you have an emergency or something, and you have to call for a tow truck or call someone to come get you, you're not just like, yeah, I'm on this highway somewhere between, Houston and Austin. If you know your mile marker, then you can actually, get people to you. With your cell phones now, you probably could use the GPS and it would tell you. Just send them a pin. I'm here. All right, so the Human Genome Project took advantage of a couple of different methods of sequencing and identifying genes. One method is called shotgun sequencing. So to do that, they take the original DNA and just cut it up into random fragments. And then they figure out the sequence of each fragment. And then they use a computer to put those fragments back together, kind of like a jigsaw puzzle where they figure out where the areas overlap. And then the computers are going to align those to the marker molecules, or not marker molecules, the marker regions. Okay, so that we know, okay, so we've found this sequence that we're using for our mile marker, right? And then um, here's where this sequence is on the DNA relative to that mile marker. Um, so it kind of ends up looking like this. They also identified and located several genes. So one thing they had to figure out, the first thing they would have to figure out to identify where is a gene at, is to look for these promoter sequences. So we talked about promoters in chapter 13, I think it was. So the promoter is where the RNA polymerase binds to start transcription, right? So that's going to be where the gene starts, is with the promoter. And so they would look for specific sequences. They're looking for the start codon. They're looking for stop codons. They're looking for regions that could be an intron versus an exon. So remember, the introns are the non-coding portions, and the exons are the portions that we actually would use for the gene, okay, to make that protein. As far as like comparing sequences, so they use DNA samples from I don't even know how many people from around the world, um, and they determined that between any two individuals, you would only have 1 in 1,200 bases that will not match. And a lot of times those differences come in the introns, and in some of the other regions where we're not coding for a gene, but you only have a difference of 1 in 1,200 bases between non-related individuals. The more related you are, the more similar your DNA is going to be. And then they can also find what they call SNPs, or what's called a single nucleotide polymorphism, which is this really sciencey way of saying that you have a single base that's different. Okay, it's these 1 in 1,200 non-matching bases would be SNPs. Okay, so sometimes you get certain SNPs together, which then they can associate that, oh, okay, these are the differences that lead to this disease or this condition or this disorder. Okay, so that was one of the things they were trying to do, was figure out what is it in the DNA that causes this person to have whatever disorder, but somebody else doesn't. So they're comparing DNA sequences from lots of individuals, trying to find where is this gene located that causes this disorder, and what is it within that DNA that is actually creating the disorder. With this, they had to actually create a whole new world of study, a whole new field, I guess, within biology. It's called, well, they actually created two. So we have bioinformatics and then genomics. So with bioinformatics, what we're doing is taking biology and molecular biology specifically. So molecular biology is studying molecules like DNA, and combining that with information science. So it's using what we're learning with the Human Genome Project and creating predictions and doing a lot of computer modeling and statistics, data analysis, using that to um, create databases and things where we can share information that we learn about the human genome and about other molecules with whomever might be interested. And then within genomics, we're studying the whole genome of different organisms, um, finding all of the genes, where they're located, what they actually do, and also like how they could be altered, how polymorphisms, right? How those differences within them can cause some sort of a disorder or other uh, non-functionality, I guess, or a different functionality. So what we've learned from the Human Genome Project is that there are 3 billion base pairs within the human DNA, but only about 2% of that actually is used to make proteins. So that's not very much. Um, Still a lot. I mean, 2% of 3 billion is still a whole bunch. They did manage to pinpoint a lot of genes, and they figured out associated sequences with, so like what is different about this person who has type 1 diabetes versus this person who does not. What's different in their DNA that causes that that individual to have a disorder or a disease. And then they identified 3 million places where we have these single base DNA, the single nucleotide polymorphisms, these single base DNA differences. Okay. So they found 3 million places of those. This is just comparing the size of genomes of various organisms. So humans have the most on here, but some of these are probably more than you would think they do. So like E. coli bacteria still have 4.6. This is a little bacteria, and it still has 4.6 million bases in its genome. Okay. This HIV only has 9,000, which only corresponds to 9 genes. Viruses, again, not really alive, so they don't need that many genes. What's interesting, or what you might find interesting, is that humans have 3.2 billion bases and an estimated 25,000 genes. But if you look down here at the mustard weed, they have 120 million bases, and they actually have a greater number of estimated genes than we do. So we have a lot of bases that apparently don't do a whole lot. This does, what we've learned does raise some issues, kind of presents us with some new questions. Some of them are ethical and might be important to you at some point. So for instance, like who owns and controls that genetic information? So if you've done like a 23andMe or anything like that, they have your DNA on file. What are the privacy rules on that? Can law enforcement say, hey, you got a DNA profile on this person? We're going to need that. Can they get a warrant and do that? Get access to your genetic information, or should that be private? So who should have access to that information? How is that information going to be used? If you've ever seen the movie Gattaca, if you haven't, I think it's one that I can recommend for the most part. So the movie Gattaca is about, it takes place in the future, and they have sequenced the whole DNA and everything. And so at birth, they take a DNA sample, and they predict like your lifespan and your risk of different diseases and disorders. And based on that, it creates this caste system. So based on your DNA sample at birth, you get placed into like you're going to be on this path. You have like not great genes. So we're not going to let you go become an engineer or something because, I mean, really, like you're probably going to die before the age of 40 anyway. So, what do you need to go study engineering for? Um, so that, I mean, and you think that that's not likely to happen in real life, but I mean, really, it's not completely out of the question. Like there are definitely people who would push that agenda. Um, there's also just an issue of insurance. If they, um, if your insurance knows that you are just predisposed to having a genetic condition, right? So your genetics could say that you are at a higher risk of having a heart attack or having cancer or whatever it is. Can your insurance company, can health insurance refuse to cover you, refuse to, or charge, or just charge you a higher premium, right? Charge you more money to be insured because you might develop this thing later in life. Can you have an employer that refuses to hire you because you might develop this thing later on? So those are some issues that are probably going to be things we have to resolve, things we have to decide. How are we going to handle the fact that we know so much more about our genetics now? What are we going to do with that information? Okay, so that should be the end of the lesson. Again, you probably need to be working on, so you have homework that goes with this lesson. I would also recommend you start on the review. Send me questions over the homework or the review. Either way, I'll get back to you as soon as I can. All right.